In comparative vertebrate anatomy, we not only name anatomical structures like bones of the skull and their subparts, but we also talk about their evolution and their history through vertebrate groups. Now, this vertebrate skull is so complex. How could one, discuss its origin? Where could such a structure possibly have originated? Well, the first principle to establish is that there are three main parts to the vertebrate skull that arose at distinct points in vertebrate history. The first part is what is referred to as the splanchnocranium, which originated from the gill arches. You may recall that invertebrates, including hemichordates, possessed cartilage tissue. And hemichordates then develop slits in their throat, which would allow them to feed better. Water comes in their mouth and leaves their throat. By the time we get to the first chordates, cartilage was actually in the gill arches as gill supports. Obviously, it's easier to keep the gills open if there was cartilage there to support it. This splanchnocranium, this cartilage of the gill arches, was the first part of the vertebrate skull. It precedes another region known as the chondrocranium, which is cartilage which would form underneath the brain, and then also around the ear as an otic capsule, around the eye, and around the nasal region. And long before the third and final section of the vertebrate skull, where dermal bone formed what's known as a dermatocranium. And so there are these three separate regions of the vertebrate skull: the splanchnocranium, the chondrocranium, and the dermatocranium. And I'd like to discuss the splanchnocranium first, given that it was the original part of the vertebrate skull, once again, arguably going back to the early chordates, which had cartilage in the vicinity of their gill arches. And so here you can see a chordate with its gill arches. Here you can see a hemichordate with its gill arches. And cartilage, once again, which is an ancient tissue, was organized in this region. So obviously, if you're going to study fish, and the evolution of fish, this becomes important. And if you were to look at the most primitive fish alive today, the lamprey and the hagfish, which are the two types of jawless fish alive today, you would see that there is cartilage in the gill arches, this splanchnocranium. Okay, so this is obviously fascinating, but why should we care about what we see in, say, the jawless fish of lampreys and hagfish? Well, these gill arches, which were made of cartilage, were modified in the jawed fishes. And so the jawed fishes modified the first two gill arches. The first gill arch became the jaws, what's known as the palatoquadrate, and the Meckel's cartilage. And the second gill arch became a supporting structure for the jaw known as the hyomandibula. So this splanchnocranium was then modified to form the first jaws of the jawed fishes. And that's obviously extraordinarily important. One could argue that no other change in the vertebrates had such a dramatic change in vertebrate positions in food chains as jaws. Prior to jaws, they were food for invertebrates. It was only after jaws evolved that vertebrates became top predators. So here you can see a shark's skull, and then included in the skull is what's referred to as the splanchnocranium. Not only do you have cartilage in these gill arches, but also the first two gill arches have been modified to become the palatoquadrate and Meckel's cartilage of the jaw, and then the hyomandibula, the structure which supports the jaws. So once again, critical in vertebrate history, where the first gill arch, which in jawless fish was only a gill arch, then was modified to form jaws, both the palatoquadrate, which is the upper jaw, the Meckel's cartilage, which is the lower jaw, and then the hyomandibular cartilage of the second arch. And plus, from the second arch, there's also a few other cartilages in sharks, which we can observe. Okay. So this splanchnocranium, important in the jawless fish, important in sharks, important in those fossil fish that we see, those placoderms, etc. Great. Why do we care beyond that? Well, because all vertebrate embryos form this throat with slits. We form pharyngeal arches, which look like they're preparing to form gills. We vertebrates who live on land, these pharyngeal arches then become modified, as in this chicken. All right, so this chicken is not going to develop gills, but as an embryo, it looks like it's going to. It develops these pharyngeal arches, and it develops cartilage in these pharyngeal arches. Here we see a pig and the same thing. So here's once again the pig, and you can see the pharyngeal arches and cartilage forms there, a splanchnocranium. What happens to these gill arches? Well, they get modified. And what happens to the cartilage within them? Well, they get modified. And we could talk about different bony fish, etc. Even things like the jaws. Much of that will form as like an embryonic scaffolding for dermal bones, which will become the jaws. So if you look at, say, a bass or a trout, they no longer have the palatoquadrate and Meckel's cartilage, the cartilage forming the jaws, but that cartilage does form, a scaffolding around which the dermal bone, which becomes the jaws in these bony fish, forms. But even so, we humans, we modify these gill arches and we lose many parts of them, to be sure. But here is a bone which is held within the middle ear of mammals like humans. And so we have these three little bones which are inside a space inside the temporal bone. And so the incus, also known as the anvil, the malleus, also known as the hammer. These are derived, here they are inside a space inside the temporal bone. These are derived from the splanchnocranium. The cartilage of gill arches then ossifies and becomes these portions of the incus, the malleus, and also part of the sphenoid bone. I'll get back to that in a second, but there's a skull bone in humans where a part of it is originating from the first branchial arch. From the second branchial arch, we get the stapes, that is the third of those middle ear bones, and also portions of the hyoid bone. The hyoid bone is just a small U-shaped structure, but it's actually quite complex and forms as multiple embryonic units come together and fuse. So this is a bone in humans which supports tongue muscles. It surrounds the larynx. And once again, forming from these regions of the splanchnocranium. Another portion of the hyoid comes from branchial arch 3. And then in our respiratory tract, the larynx is formed from separate pieces coming from branchial arches 4 and 5. So the splanchnocranium is the first part of the vertebrate skull to form. And it's important, the gill supports, very important in fish, which rely on gills to breathe. The modified gill arches become jaws, which revolutionizes the place that vertebrates have in food chains. But then finally, as the vertebrate skull is modified over millions of years, portions are retained, and we humans still retain, as part of our sphenoid bone, as part of our hyoid bone, as the middle ear bones, we still retain portions of the splanchnocranium, this original region of the vertebrate skull. The second region of the vertebrate skull to develop is what is known as chondrocranium. And you may remember that the prefix chond goes along with cartilage. So a chondroblast, a chondrocyte, a perichondrium, they all refer to cartilage. So the chondrocranium is a portion of the vertebrate skull which is composed of cartilage. And if you were to look at, say, a lamprey, which has no bone, in addition to the splanchnocranium of the gill arches, it does have a little bit of cartilage around the brain, not completely encasing it. But there are supports for sensory structures like an otic capsule forming around the ear region. When you get to a shark, and you ask, can I see the skull of a shark? Well, clearly they have a gill arch skeleton, as we'll see presently, that's the splanchnocranium. But here you can see the chondrocranium, and it's much more elaborate than would be seen in a lamprey. So it has an otic capsule region which surrounds the ear. Here you can see processes which surround and protect the eye, and there's a pedicel which actually supports the eyeball. You can see it forms a rostrum which surrounds a nasal capsule, protecting the nose. And then also it completely encases the brain. So the brain is surrounded by cartilage. Sensory structures like the nose, the ear, and the eye are partially to fully surrounded by cartilage. And so this chondrocranium now protects the brain, protects the sensory structures. Here, the otic capsule, the ear. Here, the eye would fit the nasal capsule. Here, there is an opening for the optic nerves, an optic foramen. Here, once again, you can see ridges in the area which would surround the orbit. And here you can see a pedicel, a fragile piece of cartilage which actually supports the eyeball. But just an interesting note on the pedicel. A shark is a cartilaginous fish. It's a primitive type of fish. And the bony fish of today, they don't have pedicels. But the very first bony fish, they had features that they shared with sharks and other primitive fish like placoderms and acanthodians that modern bony fish lack. So if you were to look at the skull of early bony fish, you would see features that don't remind you of the bony fish of today, but of these earlier fish, including those eye stalks, that pedicel, which is not found in any bony fish alive today. So the chondrocranium, had become more elaborate by the early jawed fishes. Now, once again, a lot of this forms kind of like as an embryonic scaffolding for the dermal bones, which will develop in the bony fish, as we will see in the next section. However, the chondrocranium is still with us. Now, this is actually a little complicated. So you could ask the question, how does bone form? And if you recall, there are two ways that bone forms. It can form as dermal bone, where just in the middle of the dermis, say of the skin, you start making bone there and the bone gets wider. So that's dermal bone forming in the dermis. And most of this skull, as we will see, is made of dermal bone forming the dermatocranium. However, there is a second way to make bone, to start off with cartilage first and then turn that into bone, okay? So you can either just make bone and it gets bigger, or you can make cartilage that then is converted into bone. And when you look at the bone of the skull of a developing fetus, you could ask the question, what kind of bone do we find in the skull? And the answer is both. So the next section of the skull is made of those portions of the skull which start off as bone and just remain bone. But then there are regions of the skull that start off as cartilage from the chondrocranium and then become bone. So in this image, and I'll focus on this in the next video, this would look a little odd to, an anatomist familiar with the adult skull, because humans as adults have one occipital bone. All right, and so here's a squama, which is made of the majority of that. But here's two exo-occipitals and a basio-occipital. We adults, we don't have that. We just have one. We don't have pieces. When we think of the temporal bone, we adult humans, we just have one, right? But in the fetus, there is a mastoid portion. Not only are there the bones associated with the ear, like the angular, the malleus, incus, and stapes, but then there's a squamous portion of the temporal bone. There's a petrosal portion. In other words, a lot of the bones of the adult are made by taking separate pieces and fusing them together. Some of the pieces form as dermal bone, and some of them form first as cartilage of the chondrocranium that then later become bone. And I'll review that again presently. So the chondrocranium, one might say, where are these pieces of cartilage, in our adult skull? The answer is, well, they're not there, but they have been converted to bone and are components of these larger composite bones. A number of our skull bones arise from taking separate small pieces and fusing them together. So a teleost fish, like a bass or a trout, like a bony fish, they have separate bones derived from the chondrocranium, a supraoccipital, a basio-occipital, and two exo-occipitals. So these are bones derived from cartilage of the chondrocranium. In mammals, these simply then fuse together as part of the occipital bone. Our ethmoid bone is bone, but it results from the ossification of the chondrocranium. Portions of the sphenoid bone arise from ossified portions of the chondrocranium, and one portion you may recall from an ossified portion of the splanchnocranium, the allospheноid. The prootic, epiotic, and sphenotic are separate bones in teleost fish, but they then form parts of the petrosal and mastoid sections of the temporal bone. And so the chondrocranium is the second portion of the vertebrate skull. And even though it doesn't surround our brain and our sensory structures the way it once did, there's no otic capsule made of cartilage, but the cartilage of the chondrocranium becomes the bone of the temporal bone, which then surrounds the ear region. Okay. A third region of the vertebrate skull developed as dermal bone, which doesn't start off as a cartilage precursor. It just starts off as, within the dermis of the skin, bone is made, and it's called the dermatocranium. This first appeared in jawless fish. Now, not the ancestors of hagfish and lampreys. They're the jawless fish alive today. So the first jawless fish did not have a dermatocranium, and they were the ancestors of the jawless fish we have today. So the jawless fish alive today, which do not have a dermatocranium, have descended from the original jawless fish, which did not have a dermatocranium. But by the Ordovician period, there were jawless fish. These would be the ancestors of the jawed fish, which did have a dermatocranium. They made dermal bone in the head, and it was these very large plates of bone. So if you wanted to say, oh, where do I see the frontal bone in a primitive jawless fish? And the answer is you don't. You see these big plates which don't directly correlate to structures which I have now. So a dermatocranium forms in jawless fish and was then present in the earliest jawed fish, the placoderms. We have these large plates of what's called a dermatocranium, once again forming over the splanchnocranium and chondrocranium. The vertebrate skull came in these three parts. Now, interestingly, if you ask these vertebrates, where did bone appear in their bodies? First, it appears as bone in the head, dermal bone. and the rest of the skeleton is cartilage. And it would be later fish which convert this cartilage, like of the fins and of the vertebral column and the ribs, into bone. So once again, these fish involve bone in the head first. Everything else is cartilage, which then becomes bone. If you ask, say, mammals like ourselves, as fetuses, how did bone form? And the answer is, in the head, there was dermal bone that started off as bone that just got bigger. The first bone that we mammals make is in our heads, like in those fish. The rest of our skeleton forms as cartilage. So we start off with cartilage arms and legs, cartilage vertebral columns, which gradually becomes bone. So we can see some vertebrate history reflected in the way that our skeleton forms. By the time you, and I'm sorry about that lower jaw, that's my bad animation there. By the time you get to the acanthodian fish, and then certainly the first osteichthyan fish, the first bony fish, not only do you have dermal bones in the skull, but instead of having these big plates that don't really directly relate to the bones that I have, now by the time you get to these fish, you say, okay, well, here are opercular bones that support the gills. I don't have those. Here's a couple little bones up here that I don't have. Oh, but look, here's a parietal bone. I have parietal bones. Oh, look at the marginal teeth. They're in the maxilla. I have maxillary bones. Oh, look, outside the orbit, there's a lacrimal bone. I have a little lacrimal bone. And so by the time we get to the osteichthyan fish, not only is there dermal bone, the way there were in the primitive jawed fish and the advanced jawless fish, but actually little pieces of dermal bone, which we recognize as being homologous to the components of our skull. And as we will see, the amphibian skull is just made of modifying the elements of the skull there. So once again, there are different ways of making bone. All right, so you could start off with cartilage and then gradually convert that into bone. And when we look at, say, our arms and our legs, that's what happens. We start off with, say, a cartilage humerus, and then, it slowly develops into bone. We have a growth plate, etc. This is what's known as endochondral ossification. And we do turn parts of our occipital bone, starting from the cartilage of the chondrocranium, we do that, make that into bone, which will contribute to the adult bone. But there then is also dermal bone, which just arises from cells in the dermis, which just start making bone and then became more elaborate. And so once again, as fetuses, we develop bone in the head first. This is a primitive jawed fish, a placoderm, which has really big plates of dermal bone in the head, like those jawless fish. But by the time you get to the very first bony fish at the end of the Silurian period of the Paleozoic era, and perhaps even the acanthodian fish which precede them. By then, you say, I actually recognize some of its bones. Not only does it have dermal bone, but look, it has parietal bones like I do, okay, here in green, okay? And so then as time would go on, as we get into the bony fish, which would be the ancestors of amphibians, we recognize, lots of bones that then I have. And as I'll discuss in my next video, as we go from the bones of a fish into bones of an amphibian, you really don't need new bones. You just have to modify the ones which were already there. So then these bones of the dermatocranium just get modified over time to turn a fish into an amphibian, into a reptile, into a mammal. And so we have homologous bones of our dermatocranium. So if you go back, to these Silurian fish, all right, like Saurolepis, you can see parietal bones, you can see a number of these pieces. By the time we get to the Sarcopterygian fish of the Devonian, we recognize more pieces. So all then are pieces which not only make up this fish's skull, but will then comprise part of the skulls of amphibians and reptiles, and then even mammals like myself. They're just modified, some fused together and some get larger and reorient, etc. Last point before I specifically start going through parts of the skull is the idea of a composite bone, as I have already mentioned. We mammals are different from the reptiles. So, when you look at reptiles, you see, lots of bones of the lower jaw, lots of bones in the occipital region, in the temporal region. We mammals, we just have one bone in our lower jaw. We just have one bone in the occipital region. See, one big occipital bone. We just have one temporal bone, not lots of little pieces. But when you look at the first reptiles, look, there's a whole bunch of bones in the temporal region of the skull. There's a whole bunch of bones in the occipital region of the skull. How did you get to the mammalian situation where there's one solid temporal bone, one sphenoid bone, one occipital bone, and not lots of little pieces? And the answer is that over the evolution of vertebrates, particularly as reptiles evolved into mammals, what used to be separate pieces then fused together. So the mammalian occipital bone is actually a composite bone made of fusing lots of separate pieces together, some separate elements of the dermatocranium and separate elements of the chondrocranium fused together to form one big composite bone that starts off as little pieces. So here's all of the pieces which are contributing to the occipital bone, both parts of the chondrocranium and dermatocranium. The same would be true of, so these all fuse together and form the one occipital bone, which is now a composite bone made of multiple separate embryonic pieces. Okay. The same thing would be true of the temporal bone. I have one very complex temporal bone here, but it's made of separate portions, a squamous portion, a petrosal portion, a mastoid portion, and then these were formed by separate portions, some of them, some coming from the chondrocranium, some coming from the dermatocranium, and then even as they fuse together, they encase the middle ear, which then has those three little ear bones here. So the platoquadrate from the splanchnocranium gives us some of the middle ear bones. So the temporal bone actually has components of the dermatocranium, chondrocranium, and splanchnocrinium if you include those little auditory ossicles, and then there's the styloid process, which also seems to be derived from the splanchnocrinium. That's a process for ligaments which support the hyoid bone. So our temporal bone is a composite bone formed from multiple pieces which squish together. And once again, all three regions of the skull, splanchnocrinium, chondrocranium, and dermatocranium contribute to the temporal bone. And then also the sphenoid bone underneath the brain also has all of these separate components. The allospheноid from the splanchnocrinium, but then also portions of the chondrocranium and dermatocranium all fuse together to form the sphenoid. So how does one get this complex vertebrate skull? And the answer is it's complicated. It took hundreds of millions of years. And what we see is, gradual stages where there are three separate regions of the skull, and they, get modified over time. And especially in mammals like ourselves, very often little pieces with separate embryonic origins are fused together to make the larger composite bones which we have.